Welcome to the 12th talk of Caps Talk series. So last time we heard how protein modeling and then it goes hand in hand with function analysis. Today we will be hearing about churning algorithm for enhanced functional annotation. Over to you, ma'am.
Thank you, Shailya. Today's talk we will try to cover on some of the algorithms that were vitally required, computational algorithms, as well as some of the kind of approaches or protocols that we had to bring up in order to better predict functions of proteins, and that's what we will discuss today. And to begin with, we are going to actually talk about 4 stories. One of them is clustering of functional groups. Another is going to be using sequence and structural motifs to annotate function in whole genome sequences, for instance. And then the third story is going to be on use of cascaded sequence searches, which we had actually discussed in the sequence theme. And finally, we'll also be presenting a protocol for searching for enzymes in genomes or transcriptomes, the way it can be done effectively. So first of all, it's about the clustering of functional groups. So the first story, it's somewhat self-explanatory. In the sense, we begin to understand where the functional sites by means of actually going after functional groups which are conserved, right? You're able to see the slides now. So the first of the 4 stories is going to be on the clustering of functional groups. So what I was going to say is that term is pretty self-explanatory. So we are going to look for conserved functional chemical groups in order to begin to recognize where the business end of the protein might be residing. And for this, let me take you through the context. And the context is pretty important thanks to the structural genomics initiative that was initially led by Professor Andrew Sully many years ago, and many biophysical groups participated in this initiative to go after hypothetical proteins, that is proteins for which no function could be annotated, leave alone their structures are to be available, but they seem to be interesting. They seem to be holding promise for either a novel fold or a new function. So therefore, crystallographers and NMR specialists had adopted one structure at a time and try to solve them. So these are all available even now. And now if you were to implore into the protein data bank, like I tried to do and I showed up in this slide, there are as many as 6,665 structures. So it's not a trivial problem. So it is also pretty embarrassing for bioinformatics people, because here it is a case of a hypothetical protein for which we even know its structure, but we do not know anything about its function. And it's quite possible, isn't it? Because now we are in the post-genomic era, so we don't get to handle proteins, we don't get to purify them, express them, understand whether it forms a dimer or which pH it is more comfortable. All that we are seeing due to this sort of sequencing techniques is the primary structure or the amino acid sequence of the protein. And even if we have the structure in front of us, sometimes we do not know what is the biological function of such proteins. All right. So what we have as an example down below, for instance, it's just a random example. It's a beautiful, stunning structure of YWQG from Bacillus subtilis, and it's a hypothetical protein. And you can vaguely see that there are 2 domains. The domain on top seems to remind us of 4 helix bundles, and the domain underneath seems to remind us of jelly roll folds, like the lectins. So perhaps here some heme will bind, and here some kind of sugar might bind. But what about cases where we cannot guess, even from the domains of individual in the structures of the individual domains? And for these sort of examples, we devised long ago during the time when I had a postdoctoral fellow from the Cambridge, after Dr. Innes had done his PhD there, he spent about 2 years in our lab, and we asked a question: given these circumstances and the rising number of such cases, can we venture to propose an algorithm, a kind of a reliable approach by which we can recognize the business end of such proteins? And this is now a different protein example. And what we have on the right-hand side is nothing but the amino acid sequence shown schematically as a line. And as ever, as talked about in the sequence theme, we exploit the information on evolution. So we go into protein sequence databases, which very stringent values as possible, so that you don't accumulate wrong entries and then accumulate instead trusted homologs. And once you have them, you do what is called as a MSA, or a multiple sequence alignment. And then you know that these homologous sequences denote what sort of chemical groups are permissible or can be accommodated, and what sort of chemical group cannot be tolerated, all along the primary structure of the protein. And so therefore, instead of worrying about amino acids, that small change that we did was to worry about chemical groups. For instance, serine, we would think of a methylene group and then an OH group and so on. All right, I have converted them into chemical groups. Then we use graph theoretical algorithms to back map on the structure. This time it's a space-filled diagram, so that we can begin to appreciate a gradation of conserved chemical groups. If the chemical groups are similar, then it's going to look red. And if the chemical groups have changed all over, when we look at the aligned set of sequences, it's going to look dark blue or violet. So it's like a weak biotic representation, so that all we have to do now then is to focus on the red parts or the orange parts or the yellow parts. And when we do this, you can see straight away that right in the middle of this beautiful other protein structure, there seems to be promise of a functional site. And this happens not once, but twice, but again and again for hundreds of families that Axel has had carefully chosen, analyzed. And he showed again and again that the conserved regions where the chemical groups are retained seem to coincide with the literature definition of what we might begin to call as an active site or a substrate binding site and so on. And that is shown in yellow circles. So that is the first algorithm and the first story that we are presenting, which is on using or exploiting the conservation of chemical groups to recognize functional sites, and simply exploits the theme that function is so important for the protein, so therefore it cannot afford to change chemical groups too much. All right, and then the second story goes as follows, for which I'm going to transit to another slide. Let me do that and then come back to you. So yeah, this is the second project or this story. I hope you can see the slides. Yes. So here we, instead of projecting and deliberating as chemical groups, we do something else, which is to recognize the functional motifs. And how do we do this? You might recall in the very first talk, I had talked about structural motifs. Yeah, the very first CAPS talk, we talked about structural motifs. And for instance, we picked up the example of the methyltransferase superfamily. And then you can see that the superfamily members looks remarkably similar. But then if you try to superpose them, put them all in the same place, there are structural variations. However, what we need to really appreciate at this point in the second story are the conserved regions, or what we call as the motifs. And for this, we would like to begin to understand the presence of these motifs, which are marked in Roman letters, in the context of the secondary structures. And the secondary structures themselves for this superfamily, it is shown schematically as triangles for beta strands and circles for some of the key and important helices. And as you can see that there are motifs all over. What does it suggest? It suggests that whether it comes to the substrate binding site or the cofactor binding site, which is called a SAM, key residues are always retained and held evolutionarily conserved because they are functionally important. And these motifs can be just followed through by means of these Roman letters. That is motif 1, motif 2, all the way until motif 10. But Here is where things can get more interesting or complicated, depends on how we view it. But what happens is you do not have just one type of methyltransferase, but you have many, many methyltransferases in nature. And even within human genome, if you were to focus, you have an alpha methyltransferase, alpha, beta, gamma, etc. And the beauty is the positions of those functional motifs are not retained in the same order. They are present, but what appears as the 10th motif in alpha will be present as the last but third motif in the beta. So you can imagine if you trust these motifs so much, how much you can get lost, not being able to annotate the appropriate function. On the other hand, if you trust these motifs enough, then you can begin to fine tune annotate function and say, yes, because the 10th motif occurs in the first place, it is an alpha methyltransferase. If it happens in the last but third place, it's going to be a beta methyltransferase and so on. So this is one example where the positions of the motifs can just rotate around and change places. And this happens because of the reason that you have in nature evolutionary changes that come under the category of what is called as circular permutation. And this is very, very simple. The N and C terminal positions do not always coincide. Otherwise, two proteins might have very similar structures. They look the same, but the N and C positions are completely different. So you can imagine how much of challenge could be there when we are handling only the primary structure of such proteins and trying to compare them. And secondly, you can have another challenge, which is something what we call as discontinuity. You can have two domains in two different proteins, but they may be discontinuous in one of them simply because there was another domain that got inserted in one of the homologs. And these pose a different level of challenge. And thirdly, you may be surprised, but it is simply true that small domains can be very, very difficult. They are notorious to handle simply because they do not have enough conserved chemical groups, they do not have enough motifs, and they do not have a high hydrophobic core, and so on. And for all these reasons, we, as users, we realize that whether you are dealing with the circular permuted domains or discontinuous domains or small domains, the function annotation of such protein domains can be quite challenging. And together we tried to consider all three such challenges and devised an algorithm which was driven by neural networks. And this was the core MSc project of one of my students, Kumar Gaurav, but he was ably assisted by a very talented trainee from IIT Kanpur who had come then, who is Nitin Gupta. And Gaurav had a biological problem, and Nitin Gupta knew nothing about biology then, but very adapted programming. So together, and of course with my guidance, we sat together and devised this algorithm, which is driven by neural networks, in which you can care for particular features, such as the presence of the motifs, the order of the motifs, and the intermotif distances. And that could be a default. However, the beauty is, since this is through neural networks, you can simply just switch off some of these parameters. For instance, you can say, I only care about the motifs and the order, but I'm not bothered about the intermotif distance. And if that happens, then you can handle certain cases. And for instance, if only the motifs and the intermotif distances are considered, you can actually approach discontinuous domains rather well. And if you're looking at only the motif and the order, you may be able to look at some of the circular permutation well. And if you're actually looking for only one or two motifs and not the distance or the order, then you may be able to handle small domains well. And this exactly like how you switch off in an electricity board, you can just simply switch off some of these parameters, and then or which parameter you will like to switch off depends on the type of challenge you're facing, whether it's small domain or discontinuous domain or a circular permuted domain. And then you can tune your algorithm and start the search procedure. So it is a very simple idea, but I hope you appreciate that it can be pretty useful depending on the type of challenge that you anticipate and then use those sort of properties to then gauge your function annotation predictions appropriately. So all this was contained into this algorithm, which we called as FSSM, which stands for Functional Annotation in Whole Genome Analysis using sequence and structural motifs. And this particular algorithm works quite well, and it's comparable, and quite often in many different families of variety of folds outperforms other sensitive algorithms such as HMM and something which is called as Impala at that time. So this is the second story. I hope you liked it, you appreciate it, and in fact, I would be more than happy if someone can adopt it once again, and then we can compare it with the current available methods, or we can add more features and so on. So that was about the second story, right? So first was the clustering of functional groups, and the second was to go after sequence and structural motifs, drive it through neural network, and then see how best we can then begin to attribute very fine-tuned appropriate functions to newly recognized protein sequences. Next, the third and the fourth stories are going to be presented by others in the group or alumni in the group. And first, the third story, therefore, is going to be of cascaded sequence searches. So, right, you just give me 1 minute. I'm going to close this and then come to just a few. If Sandhya can give me some time so that I can introduce the topic, it will be good. Let me try that once again. So we have the cascade sequence search, and within that, I'm going to just very briefly introduce the topic. Right, you're able to see the slide? All right. Again, this is about a protocol that was established many, many years ago and carefully and very fondly followed up by late Professor N. Srinivasan. And the idea is simple. It borrows the idea proposed by late Professor Cyrus Chotia on the powers of intermediate sequences to then connect two neighboring families. And this is very important again, because unless and until we are able to train our sequence search algorithm, are you able to see the video now? Yes. Okay. So unless and until we train the sequence search algorithms to appropriately and efficiently recognize two proteins with mere sequence information, how are we able to then use it for functional annotations, right? So if two families are indeed functionally related, our sequence search algorithm should be able to recognize that better. Yes, for this, Cyrus Chotia had proposed that the intermediate sequences can help make these connections, and late Professor Srinivasan had collaborated with our group and showed that you can use simple SciBLAST to then take this through generations of sequence searches, which will enable intermediate sequences also to get a chance to go into the sequence search arena. And in doing so, you have the possibility that you can begin to make connections between two protein families. And that's not all, because sometimes you can happily connect up using these sort of approaches, but where two families fail to get connected, we then decided to generate what we call as artificial sequences to then bridge the gap between two families. And this seems to work well for most, but not all. And therefore, the project then extended to look at two families which fail to get connected to together generate an artificial sequence, which then has the possibility to connect difficult families. And this can be quite fascinating as well as complicated, because then you can imagine a whole fold, many different families and superfamilies all have their own crisscross connections. However, the hope is that the function annotations become meaningful still and easy with mere sequence information. And here, for instance, typically it involves mathematical models of two different families under question, and also to hybridize their mathematical profiles to then carefully generate the artificial sequences. These were all done very nicely by a combined collaboration with Dr. Nagasoma Chandra's lab. in the biochemistry department of Indian Institute of Science, along with one of her students, Richa Mudgal, as well as Sandhya Shankaran, who was the lab student, CAP student, and as well as Gayatri Kumar, who particularly focused on the generation of artificial sequences and following them through the sequence searches. So Sandhya is going to present this third story. I request Sandhya to make her presentation. Please wait.
Sure, ma'am. I hope you can hear me. I'm just about to share my screen. Just a second.
Yes, we are waiting. Yeah. We are able to see the screen. Perfect.
Okay, all right. So very quickly, I mean, ma'am has given you a primer on why we need to do this. So primarily, we had started looking at several cases where we had started seeing in many of our sequence search efforts that for a very large number of protein families, although sequence identity is very, very high and appreciable. And therefore, when we use any sequence search method, then it is very tractable and easy to connect these proteins. However, for proteins doing the same function, we also started noticing that sequence identities can be really, really low. And not sure why, yeah. And in fact, for the proteins doing the same function, sometimes it can even go as low as about 11% sequence similarity. So when we start capturing the nature of connections that you can easily make through, say, a simple search method like BLAST, and for example, I've taken the representation of the alpha toroid fold here. The lines in black show that if there are different members within a particular fold, these members can be at the level of protein family or at the level of protein superfamily. But that notwithstanding, sometimes the sequence search method is simply able to find the connections because the members have as high as about 90 or 60% sequence identity. But when this starts falling and it's about 11%, it is not unsurprising, therefore, that you may have such lone clusters, right? So you may have some blobs which are not connected, for example, at all. And when we try to represent that dispersion of sequence identity within the protein family, then we see that maybe they exist as islands which are unconnected, right? And therefore, at that point in time, there was this. Are you able to hear me? Yeah. Yeah. So at that point in time, as ma'am mentioned, there was a lot of interest on the intermediate sequence-based searches that were started in Professor Cyrus Chotia's groups and continued by several others, where they noticed that there were some proteins which can have the properties, sequence properties of both the query and the distant hit. And this is especially useful when the sequence identity is very, very low. So our work really started looking at using an existing sequence search method and powering through sequence searches in a very rigorous manner. At that point, SciBlast was very, very popular when this program was originally written. And therefore, we thought we would test it on the SciBlast platform. Here, what was done really was that starting with a single query, if you were to search within the database in a typical search protocol, we would stop here. We would be happy with what hits we would get at the end of this search and examine how similar or close they are to the query sequence. But if you really want to tap the potential of distant sequences, then we decided that we would look at the sequences which come up as hits. Take each one of these hits and then propagate another round of SciBlast searches with each of these hits, which will now become queries, and look for some more. So this process of iteratively performing SciBlast using the hits which we get at the end of every generation, what we call as a generation, it helped us to really move through protein sequence space and jump from one natural linker to another natural linker and so on and so forth. And therefore, the original islands which were disconnected, we would be able to connect them simply by tapping the potential of the natural sequences. So the original algorithms were tested on the Pali database, but subsequently tested on several other databases as well and found to be quite generic in application. And one might wonder how we tested it on a large number of protein families. So just to show you a sample, we would start with all members of the TIM fold, and we would take each one of them as queries in a SciBlast search within the PaliPlus database, and then identify homologs, take each of the homologs and loop back and search again in the database and keep going until we would not get any more hits. So you might easily appreciate that these are very, very rigorous searches, and they can be a bit time intensive depending upon the number of queries that we want to begin with. And at those, when we started this analysis, it did take initially a large amount of time. But what was nice at the end of this entire exercise was that where we would test the performance or the ability to improve on the number of connections that you would get in a regular sequence database search, we found that we were able to improve the coverage by as high as about 15 to about 30% for many of the protein families. And indeed, when we look at protein superfamilies, we were even more excited because there were some here, such as the alanine racemase, for example, where we see a very, very nice scale up of about 10% coverage to about 82% coverage, or 4% coverage in the case of the rhamnose isomerase family, and going up to about 78%. So a lot of excitement in looking at the power of tapping the potential of natural linkers, right? So this graph simply shows you a performance and the improvement in coverage. And what was nice at the end of this entire exercise is several protein relationships that, especially when we look at members within a fold, we are able to recognize these relationships only when structures are available. We found that this approach found many of these connections by simply using a sequence search method in a very rigorous manner. For example, the relationship between mannate synthase and 2-dehydro-3-galactoride-dealdolase, which are all members of the same same fold, we were able to find some of these connections. And when we look at the residues that are conserved and so on, obviously, there is a lot of meaning to it. And since they are members, known to be members of the fold as well. And likewise, there are some relationships which we were also able to detect, which were beyond the fold, moving beyond the fold. And here, our question was, are we looking at false positives? And but literature seems to support the view that for many of these, which technically we would call as false positives, simply because they are connections across the fold, there is reason to believe that there are some very, very nice similarities at the gross structural level. And a sequence-based search method being able to capture this lent us a lot of excitement at that point in time. And of course, we wanted to translate this into something which everybody could use. And we developed a web server for this called the Cascade SciBlast web server, which has been hosted. And I am happy to say that the version which is currently hosted in the NCBS is still a working version of the program, where the user has the opportunity to submit a sequence and look at the hits at the end of every generation and select the ones with which they want to propagate the further generations. So having said this, the use of the server helped to establish several relationships which are really not easy to appreciate unless you have their structures, such as the ones between kinesin and myosin, as was described in this paper. So the power of Cascade SciBlast clearly in propagating lent itself to a lot of excitement by itself. And we moved forward from here because we realized that in being able to connect protein families, which are represented by these blobs of sequences, one potential is to use natural linkers to move between the blobs. But there is a possibility that some of these natural linkers may not exist simply because they have been selected out during evolution, because there are mutations which are constantly acute, but not everything being very stable, or they simply may not have been discovered. In such cases, we decided that we might want to generate artificial sequences. And obviously, how would you generate such an artificial sequence? Well, natural linkers somehow inscribe the properties of both the proteins that they are trying to connect, right? So protein A and protein B are distant, having a low sequence identity. And therefore, a natural linker may have a high sequence identity with either one of them. Why not use the same logic in the design of artificial sequences? So here we thought one of the ways, and this is work largely done by Richa Mutkal, is to use a directed sequence design procedure to connect protein families. So starting from protein family A and protein family B, if you can represent or capture these protein families as profiles or mathematical models, as ma'am also mentioned, then a marriage of the two in the form of a profile alignment can help to create a model which will probably represent the natural linker population. And we adopted then a roulette wheel based procedure to randomly synthesize designed sequences which or computationally generated artificial linkers to try and connect protein families. So when we did this, obviously, we tested it against various parameters, and we found that in going from the design of sequences from protein families, one can look at a very, very natural way of propagating or improving the number, the dispersion around the protein family members. So in this case, for example, in the case of the ubiquitin-related family and the UBX domain family, which we know are related after looking at their structures, they are still fairly distant in sequence, as shown by the representation over here. But if we were to design sequences in the method that we developed in the lab, then we were bridging the gaps in this space. And this is also captured here through this cluster representation, where the blue and the greens indicate the distance and the purples indicate the designed intermediate sequence with which we populated the sequence space. So I'm showing this pictorially, but we went on to then create a database of such artificial linkers. And this database is referred to as the enriched data. And so in the enriched database, we had a large number of these design sequences, and we clubbed these design sequences with naturally occurring sequence databases, such as the SCOP database. And as in the case of the cascade SciBlast, we performed assessments by comparative analysis of performance of searches in the database, the original database, which did not have any design sequences, and the other database, which had design sequences included. As in the case of the cascade SciBlast, here also we saw a powerful jump in the number of protein superfamilies that we were able to connect, as I have shown here through these graphs, and also through this sort of a network picture, where the original clusters which were distant suddenly became connected when we included design sequences in our search. How well did we perform in any method? We look at it in terms of specificity and sensitivity. We did very, very well in terms of the sensitivity, as you can see in the plot. Therefore, one of the applications of this is to look at domains of unknown function, which everybody was battling with and trying to find or associate a protein function to. And here, when we tried this on a very, very large number of DUFs, about 614 of these, we found that for many, we were able to make associations with a known fold. And so one of the examples that I thought to highlight today was that of DUF 1572. And which we noticed were from diverse bacteria, and a fold could be associated using the enriched database searches. When we look at the basis, the functional or the biological basis of why we might be making these connections, and look at the structural superposition of a member of the DUF protein and that of the known fold to which we were able to associate it, well, in this case, we were able to get quite close to prediction of a potential binding site. We found many of the residues which were implicated in the function of that particular protein family were also retained in the DUF member. So clearly a method which was powerful simply by starting with natural linkers could be augmented by using more and more design sequences, which we call as artificial linkers because they were generated artificially. So with this, I will end and maybe move forward to the next presentation.
Thank you so much, Sandhya, for a very lucid presentation and also explaining the kind of approaches that are available, and especially around the cascade sequence algorithms that can enable better function annotation. Yeah, thank you so much. Please stay on. And then next, what I would, if I may use the screen now, I would like to introduce to you all the fourth story, which is on the choice and the development of a protocol for searching purposely into a genome or a transcriptome assembly data. And this will actually become more relevant as we go along to the next theme. Right now we are in the function theme, but next theme is going to be on the medicinal plants and how one might use genome or transcriptome data to better recognize functions of genes embedded in them. And here we are asking the question, if we have an enzyme of our interest, and we would like to implore into either genome or transcriptome data, how do we do this? And this is now like looking from one known to many unknowns, because the entire genome transcriptome we assume, let us say it's new, we don't know much about it. All right, so there are more questions than answers. So for instance, one might ask which method to use. And I must say, although the story is going to be presented later on, this genome or assembly or the transcriptome assembly is going to be provided in the form of what are called as scaffolds, which are bits of the chromosomes. So if we catch hold of one of those scaffolds, which method are we going to use would be a question, BLAST or HMM. And then how do we recognize the appropriate scores in which the right connections are then firmly determined? And what do we do if there are partial genes? Because after all, we are looking at chunks of the chromosomes. So, and the genome itself may be draft quality. So how do we do that? And also, how do we assign different thresholds? Like any other sequence search, we'll have to standardize and come up with the appropriate working evaluate threshold, because we do not have a very huge database. We have a genome of interest. So the evaluate threshold also has to be appropriately standardized. And this then relates to even an HMM search. All the values have to be appropriately attributed. Plus, there are additional questions. For instance, one will begin to wonder, what are all the queries? What's going to be our start point? If we are searching for an enzyme, do we stop with one representative, or we should take as many as representatives as possible? Should we give it as a jumpstart alignment, the way Soni Malhotra had explained on the DNA binding protein families, or should we take in one of them? And Advait Joshi is going to present the story. And this did not happen so easily, what the protocol he is going to present, because we did have a number of discussions, debates, and arguments. And in fact, this relates to the next theme, which will be presented later on. But at the end of the story, he will present, he will give you a glimpse of the people involved, and you can easily imagine how complicated it is to arrive at a consolidated solution. We decided, for instance, do a phylogeny and then recognize which queries will go in for searches, and then definitely do a literature survey so that we begin to know which are the business end of the proteins, namely the functional residues. And after doing the sequence search into the genome of or transcriptome, we might accumulate hits, but like how Sandhya had presented, what if the hits are so broadly picked up because of the sensitivity of these sequence searches, that we are picking up not the correct member, but many members across a whole superfamily. So what do we do in that case? Again, do phylogeny, look for functional residues, and if you have any proof on transcriptome data, better employ them. And that's how this protocol then evolved. So this is what Advait is going to present. Advait, please. So this is our fourth story for today.
Thank you, ma'am. Thank you for nicely introducing the topic. Hope my slide is visible in the presentation mode. All right. So ma'am has nicely primed you all about this CAPS protocol, and how in today's era, where we have a lot of genome sequencing projects going on, how to get annotation for these newly sequenced genomes, transcriptomes, and even look for making sense out of this genomic data. So one of such an attempt is this CAPS protocol, where it is a knowledge driven protocol, and we have emphasized on the proteins involved in biosynthetic pathways, and I'll show you how we go ahead with this CAPS protocol. Just a second. Yeah. Okay. So first of all, so if we have a newly sequenced genome or a transcriptome, and we have a metabolite of interest in our focus. So what we do is identify the pathway involved in biosynthesis of this metabolite. There can be several resources like EcoSeq, PlantSeq, where we can identify the pathway involved for the synthesis of this metabolite, along with the enzymes involved in this pathway. Now, when we have the enzymes, we can either get the EC number, or if not, we can look for such enzymes in sequence databases, where we can find several homologs for that particular enzyme, not necessarily from the plant of interest, but it can be from several other related plants or even distantly related plants. Now, when we have all such sequences, we have to curate this data set so that we start with a very good set to search in our genome. So the curation of these sequences is done in two modes. One is we align them as a multiple sequence alignment, and then try to identify whether the functionally important residues are mapped over there. So functionally important residues, or the FIRs, they relate to the binding sites or maybe active sites. So there are several such residues which are typically provided with annotation in sequence databases like UniProt and CBA, and then we can identify such residues which are key residues, and are we finding them in this multiple sequence alignment. Along with that, we do a phylogenetic tree reconstruction and see whether within these sequences, which are our start points, are there any outliers. So like here you can see the fifth sequence can very well be eliminated because it does not have a FIR map, and it is also not clustering with the known sequences. So this is how we curate our starting point. And using this multiple sequence alignment, we construct what is called as a position specific scoring matrix. Now we just heard from Sandhya about the power of PSI-BLAST. So this PSSM is what drives this PSI-BLAST. And what we do is construct a PSSM from this multiple sequence alignment, and then follow a protocol in PSI-BLAST called as jumpstart PSI-BLAST, using which we query our proteome or our genome of interest. And once we search this proteome of interest, we get a bunch of hits. Now again, these hits we need to validate whether they are true hits or they are sequence homolog, but not necessarily exactly the same protein which is involved in biosynthesis of the metabolite of interest. Again, we follow the same two-way schema where we do a phylogenetic tree reconstruction as well as FIR mapping, and that's how we can select which are the true hits and exclude which are not. Further to this set, we can validate using experimental procedures such as QRT-PCR analysis or any other experimental validation, as well as some computational techniques which I'll come to in the subsequent slides. So this is the entire CAPS protocol, and using which we can easily try to relate or get trusted homologs for the enzyme which is involved in the biosynthetic pathway of a metabolite. So to narrate about the CAPS protocol, or to take an example, I'll navigate through the Moringa transcriptome work. So in Moringa oleifera, the drumstick plant, we perform transcriptome using 5 different tissues, that is flower, stem, pod, root, and leaf. And there are several metabolites which we were interested in, which we wanted to chase. And for this, we implemented the CAPS protocol. So among these metabolites, this is a typical picture you can see in any transcriptome studies where there is a relative abundance between the tissues that have been used and the transcripts that were identified for such things. But how we identified these transcripts? So I'll navigate using example of a metabolite called as quercetin, which is a flavonoid, which is attributed with antioxidant and anti-inflammatory properties. Now this is the quercetin synthetic pathway, which contains 7 enzymes, and we identified 9 transcripts for this. So in interest of time, I will talk about one of the enzyme involved in this quercetin pathway, which is chalcone-flavone isomerism. So how we identified the transcript, which is responsible for this step. So if you see here, this is the same schema what I presented earlier for the CAPS protocol. So this is the pathway. There is this enzyme chalcone-flavone isomerase. Now we search for these homologs of chalcone-flavone isomerase in the sequence databases. We also take help of the enzyme commission number, and we get a bunch of hits. Now these are among these hits, we have to again curate the input set. So for curating this input set, we use the same technique, that is FIR mapping as well as phylogenetic tree reconstruction, and then select that some of these hits can be part of that PSSM, which we use for jumpstarting SciBlast and search in the Moringa transcripts. Now in this, again, we get a bunch of hits. Among them, we select this as the best hit because it has FIRs mapped onto it, and also it is co-clustering with the known homologs. Further, we validated this transcript by QRT-PCR, and also it alludes to the point in the larger schema of transcriptome study that the relative abundance across tissue was observed in the same fashion as observed in QRT-PCR expression levels. Now, we compared this CAPS protocol with several other publicly available tools such as CAS, which is cake-based, WebMGA, and Mercator. But what we found that, say, for the quercetin, along with quercetin, there are several metabolites that we looked. In fact, we looked at 10 different metabolites, which corresponded to 37 candidate genes for the enzymes involved in the biosynthetic pathways. Now, among them, Mercator performed very well because it is a plant genome-based annotation pipeline. CAS performed like 89%, and WebMGA did not fare well. But the interesting point was, the point of contention was that these are within the brackets, you can see these are the extra hits which they pointed to. And these were filtered by CAPS protocol because we could easily identify the FIRs which were present or FIRs which are absent in the extra hits. So that's the power of CAPS protocol, with which we can easily identify the trusted homolog. Now I'll also discuss one more metabolite, that is vitamin C. So this yellow part is the vitamin C biosynthetic pathway, which is typically seen in plants, and the blue ones are found in animals and other organisms. But among this vitamin C biosynthetic pathway, we focused on last 4 steps. And in this, I will talk about the last enzyme, that is the GLDH, which converts the galactonolactone to ascorbic acid. So this enzyme is localized in mitochondrial inner membrane, and it is also part of respiratory subunit complex. So what we did, we not only identified its transcript or the transcript for in the Moringa transcriptome through the phylogenetic tree reconstruction, and we also tried to see the substrate binding motif present in this hit and ascertain that this is a true hit or a trusted hit. Further, we also went a step ahead in the validation, where we could build a structural model of this protein, and we could, using Autodoc, we could see that the substrate binds specifically in the site, and it also made us to comment about proper substrate binding, whereas there were some predicted substrate binding which were lying far apart, as you can see over here. So using CAPS protocol, what we achieved is a identification of trusted hits and pinpoint to the correct homolog, whereas exclude those which get picked up through sequence searches but may not have the functionally important residues mapped onto them. So this was talking about the Moringa transcriptome, but we have used this CAPS protocol in several of our labs genomic projects, one of which is the fenugreek transcriptome, where again fenugreek, that is Trigonella phonum graecum, which we used for transcriptomic study using the leaf, root, and stem tissue. So it also contains several secondary metabolites which are implicated with several medicinal properties. Among them, trigonelline, which is attributed by the name of the plant itself, Trigonella, is a nicotinic acid derivative which is attributed with hypoglycemic, neuroprotective, antibacterial properties. So it is synthesized using an enzyme called as nicotinate N-methyl transferase. And this enzyme, we could identify in the phenogenic transcriptome and also validate it through both the schema as I explained earlier, that is phylogenetic tree co-clustering. So it co-clustered with a medicago truncatula protein. I mean, you may not be able to view it. Just trust me on this, that it co-clustered with medicago truncatula, which is the same Fabaceae family plant. And we got several hits in the phenogenic transcriptome. But these 12 hits did not have the functionally important residue mapped over here, which is one of which is substrate binding motif aspergine 21. So this is how we can pinpoint to the actual hit for that particular enzyme. Now, not only this, we also sort of the CAPS protocol was designed, or its inception happened when we actually started working on Tulsi genome, and over the time it developed, and we could publish it along as a companion article with the Moringa transcriptome. But over here, I want to allude that even in Tulsi genome sequencing work, we saw several metabolites like the ursolic acid and alpha amyrine, several such metabolites. But apart from that, I want to highlight on the fact that we followed up the Tulsi genome sequencing work and used CAPS protocol for other proteins as well, one of which is a rhomboid protein, which is a transmembrane serine protease. And this rhomboids, they have several types like secretase, par type, mixed, and inactive type. And using the schema, what we use, that is mapping of the functionally important residue as well as phylogenetic tree reconstruction, we could see how well different types of rhomboids, like we obtained 22 hits, and all such hits could very well be segregated into different types. So that's the power of CAPS protocol, which can be used for not only the enzymes involved in metabolic pathway, but even for other proteins as well. So to summarize, it is a knowledge-driven protocol where we obtain all the known hits and curate them, and it again employs this sensitive profile-based sequence search, which considering the diverse evolutionary context is better over a single sequence. And it is complemented by a very strong validation schema, which has a phylogenetic tree reconstruction and co-clustering along with mapping of FIRs. Now, despite all this utility, it does have a limitation that it needs this manual inspection. Thereby, there is a lack of automation, but still it is a very robust platform or a robust protocol to know or mine for such proteins in our query genome or transcriptome. So this is the team of CAPS protocol. It actually is part of the Moringa olifera transcriptome study, where it was published in Genomics, and CAPS protocol became a companion article in Methodx, and the data in brief also was another companion article. And these are the current members and past members for team CAPS protocol. But as I mentioned earlier, its inception was since the Tulsi genome sequencing project began, and we are using it for several other plant genomic and transcriptomic work in the lab. And I would like to thank several of my past CAPS colleagues who were involved actively, as ma'am mentioned earlier, that we used to discuss about what should be the correct way, and that's how it developed into a nice protocol. As well as that, I would like to thank US for giving samples for all this plant genome sequencing work, CCAM for mass spec and its PLC facilities, and certainly the funding from NCBS and Bioinformatics Center grant awarded to Professor Sardamini, and the JC Bose Fellowship awarded to Professor Sardamini. And with this, I'd like to thank you all for patiently hearing this. Thank you. I'll end my talk here.
Thank you. Thank you.
Over to you, ma'am.
Thank you. I still want to say a few things more. I think this was a very important turning point in our understanding of the computational searches, sequence searches, and fine-tuning it for looking into genome and transcriptome data. So this is what we call as a CAPS protocol. But today we had presented 4 stories. And the first of them was a clustering of the functional groups, which was done, as I said, by Dr. Axel when he was a postdoctoral fellow in our group. And this was along with actually another person. But now Axel is a faculty in one of the universities in France. And second story was on FASSM algorithm. And both Kumar Gaurav and Nitin Gupta had worked on the development of the algorithm. And now I know that Kumar had done his PhD in University of Sydney under Professor Shobha Ranganathan. And I know also that Nitin Gupta is now a faculty after he drifted to neuroscience. Now he's been absorbed in Indian Institute of Technology, Kanpur. And then in the third and the fourth stories that were on the cascade and the CAPS protocol, the number of people were quite a few, but in particular, Sandhya Sankaran, who had presented the story today online, is now the head of the Department of Biotechnology in MS Ramaya University, quite close in Bangalore. And whereas Dr. Gayatri Kumar is a bioinformatics specialist in MD Anderson Cancer Center in the USA. And Advait Joshi is currently with us, but he's in the DBT Bioinformatics Center grant. And he had presented the CAPS protocol today. And with this, I hope you had liked the 4 stories that we had presented today. And also, I would like to request Mohammed Shafi to quickly present the publications that are corresponding to these 4 stories, as well as to announce the title for the next talk next week. Until then, please stay tuned and please take care of yourself. We'll meet you next week, Thursday at 3 p.m. Over to you, Shafi.
These are the relevant publication related to today's talks. And the first set CFG analysis, ma'am talked about Dr. Timi's work. And the second one, FASSM work, Kumar Gaurav's work, ma'am again talked about that work. And Sandhya talked about cascade searches, and she talked about enriched database and other 2 work from IIC. Then the last one, they talked about CAS protocol. So coming to the next week, in CAPS talks, we have another set of interesting topics, and we have titled them Cellular Assays, Dynamics, and Ligand Vehicle Screening Towards Understanding Protein Functions. Please stay tuned.
Thank you, Shafi. And thanks to the audience as ever for your patient listening, and I hope you enjoyed them. Thank you.